Now we will discuss some of the techniques which are conventionally used in molecular biology for separating the molecules, proteins, nucleic acid and enzymes. And these all techniques, they are chromatographic techniques. Today in this module, what we are going to discuss is an important technique known as the partition chromatography. Now, partition chromatography is based on the difference in the retention factor, which is K, and distribution coefficient known as the KD. And this KD and distribution coefficient, they are of the analytes. Analytes are the compounds of which we are going to analyze, and they are using liquid, stationary and mobile phases. You know, in the case of chromatography, there are always two phases. One is the stationary phase and another is a mobile phase. Now, partition chromatography is subdivided into two classes. One is liquid-liquid chromatography and another one is the bonded phase liquid chromatography. These are the two classes of the partition chromatography which are used. Now, liquid-liquid chromatography is that in which the liquid stationary phase that is attached to a supporting matrix. There is a supporting matrix onto which the liquid phase is attached by purely physical means. What we mean by purely physical means is by adsorption or by any other van der Waals forces or things like this. They are not chemically. And the bonded phase, the liquid chromatography, is in which the stationary phase, as a matter of fact, is covalently linked to the matrix. The matrix which is in the column, with that the liquid phase is attached by covalent bond, by the chemical bond which is more strengthened. Now, an example of the liquid-liquid chromatography is the one in which water is a stationary phase and is supported by a solid phase which is a cellulose, starch or the silica matrix. These are the common supporting materials which are used in the chromatographic columns. Now these, that is the cellulose and silicon, they have the ability to physically bind as much as 50%, that is the weight by volume of the water, and remain free-flowing powders. That is what happened, that with the silicon, cellulose and all that, 50% water phase that can be absorbed, and even then they will be a free-flowing powder that don't coalescent, that don't adhere to one another. Now, advantages is that these materials, they are very cheap and they have got the high capacity of absorption, and the second is they have got a broad selectivity of the chemicals or the compounds or the molecules of which we want to separate. Wherever there are advantages, the disadvantages is that the elution process. Elution process is that to remove the analyte which we have absorbed on these columns or on these supports, to remove them, that is called the elution process. The elution process may gradually remove the stationary phase. That is when we elute the chemical, elute the molecule attached to them, gradually then those chemicals or those supports, they are also eluted or they are diluted, and thereby they alter the chromatographic conditions. Of course, when these supports, they are being removed slowly, then the chromatographic conditions in the system will change. Now, how to overcome this problem that this stationary phase shouldn't be removed? That is by the use of the bonded phase, and this explains their more widespread use. That is, we use then a support which is bonded, which is attached to the molecules, and that can be more widespread. Now, bonded phases, they use silica as the matrix. Instead of cellulose, there is a silica which is used as a matrix, as a support, and then this derivatised to immobilise the stationary phase by reaction with organochlorosilane, a compound. So this is bonded, that the silica is, as a matter of fact, bonded, attached to the organic chlorosilane, and with that it becomes, or it provides a support which cannot be moved by the elution during the elution process, and the chromatographic conditions of this our column that remain the same, that remain the unchanged, giving a similar uniform results.